starts from the recognition of need. So recognition of need and how it can be captured. So recognition of need means that first of all, you must know before investing into the plant that which products you are interested in or which products are feasible for you to manufacture and market in the region which you are targeting. So, for example, there are various products. They are OTS products, they are cephalosporin products, they are penicillin, they are CNS product, hormones, aerosols. So, first of all, you must know that which focusing. So, how you going to know? Companies who are renting their services for market research. So invest in pharmaceutical
यार
manufacturing
रिजवान
so they
इधर
can
आओ
view in which
ये
molecule,
ढूंढ रहा
in
हूँ, वही
which
मिल गया.
product, in which group setting you should work for. So they have the data and they share you the data and on the basis
कैसे हो
decision based on your market
यार
competency or based on your manufacturing competencies or based on your corporate competencies. For example, if you are taking a very tricky or very complex molecule, then you might face the manufacturing challenges. But if you are taking a very simple molecule, then you might face the marketing challenges because market is not allowing you to position yourself inside. So it's all a trade-off. So you have to be very careful while you are selecting the product. So how you can select the product once you get the data from the marketing research or historic data, then you will list the data in the form which I am showing you. This is this is this is one of the uh, project which we undertook during the last few years. So this is the data. Product classification. Product classification means a product code, unit pack size, state of dose, packing form, packing material, whether you are, this is the primary packaging material, whether it's a blister, is a glass vial, or it's infusion. So dosage form, you are what is inside the unit pack is a solution for injection or something else. So then it's a sales data, is a area classification. Area classification, for example, what is the requirement of the area class of this product, this molecule. So the area classification is there. This area classification is C. Then terminal sterilization, if it is a injectable, then whether you go for the terminal sterilization or aseptic filling. So, and rest is the hygroscopicity of the molecule, the first year forecast, first year volume, batch size, and the other marketing things. But the very important thing is that you must know that how many years you are going to lasting with your investment. For example, if I told you that I need the investment for the next 10 years or 20 years, so you have to, on the basis of your sales forecast and your growth rate of the product, you have to find out the appropriate equipment. So nowadays, it is better to over invest on plant. It is better to rather on over investing the plant. It's better to uh, go for the modular plan. So modular plants are the plants which are expandable. They have the flexibility to expand according to the sales demand. So it is better to go for the modular plant. So the modular plant are now based on clean room panels. You just develop a gray structure, and the rest will be done by the clean room panels or flexible buildings. So nowadays it is a very common trend. So I think it is very smart trend as well that you go for the clean room plants or clean room panels instead to be on the to build a rigid building with the traditional buildings like with cement and masonry block and concrete and everything. So, product classification like beta-lactam, cephalosporin, hormone, CNS. Product code is commercial parameters, SKU number, unit pack size, number of primary packs per secondary pack. So this is all the data you must gather while you are developing your own facility. Able to find out how much investment you are going to inject in your project. So how much investment means, for example, in pharmaceutical industry, I usually recommend to invest not more than 5 to 10 years. So after 5 years, the current GMP practices are starting to change. So it is better to invest for the 5 to 10 years. So after 10 years, you will be in a position to completely get back your money with profit, and then you rebuild the plant according to the new GMP guidelines or new regulations. So, for example, if it is a textile factory or if it's a automobile investment, then you can do the investment for the next 20 years. But for pharmaceutical, where the regulations are changing with the respect to months or with respect to years, so it is better to safely invest not more than 5 years. So this is one of the example I've shown you that this is the table. They are almost I think 83 products. 83 products are I think 83. There are 83 products which the investor is willing to manufacture inside the factory. So we have developed that plant for 83 products. So after developing this picture in Microsoft Excel or in Python and analyzing the data you get from the marketing research firm or historical sales data, you will be in a better position to invest to find out that whether you will be going for the cephalosporin product or you will be going for CNS product or you will be going for OTS product. So then after, you will develop a macro view, the holistic view of your plant design. For example, let's take an example that we are stuck up with the very simple like OTS products. So for OTS products, we are developing the flow diagram or process flow diagram. So you can very well imagine that how much effort you are going to insert while investing in pharmaceutical plants. So I'm showing you the process flow diagram. So this is a typical process flow diagram. Invitation of business trial. If you are the investor, so you are sitting here. Then you hire a pharmaceutical consultant. Then after hiring the consultant, you hire a firm for marketing research or you get the data from the market by your own sources. It's up to you how you gather the data. Then shortlisting of molecules based on the data you sought and analyze and find your competencies as well, that whether the selected molecules are easily handled by your competencies or not. So it's a trade-off between the competencies and the marketing revenue. So if you select some complex molecules, then definitely you will get a good return on investment. But if you are getting very simple molecules, like OTS molecules or even cephalosporin molecules, then definitely it depends upon the market saturation whether the selected molecules are capable to penetrate or expand into the market or not. So for complex molecules, it's easy to position yourself in the market, but for non-complex or simple molecules, it's really tough for you to position yourself into the market. So after sorting molecules, you go for the demand forecasting. So you, as I told you earlier, that the demand forecasting based on the 5 years investment. So you must know that how much volume of these molecules will be handled after 5 years. So it depends upon the demand forecasting. And then after demand forecasting, you develop the pricing strategy for each product. And on the other hand, you will initially estimate the resources. You must know that in order to fulfill this demand forecasting till next 5 years, how much resources I am needing. For example, how much area of the plant, how much equipment, what are the equipments, what's the size of the equipments, and what will the size of the utilities, like what's the size of the HU, what will the size of the water system, and other amenities. So after initial resource estimate, you will go to the resource data analysis. And after resource data analysis, you will having a quality function deployment and risk assessment of the design. So this is something which is a part of the quality by design. So what you are doing, this QFD or risk assessment, because conventional designs are not containing the risk assessment. In conventional designs, they are totally based on the regulations and the set standards of the industry. But in risk-based designs, you are anticipating, you are assuming all the possible risks, and then after assuming all the possible risks, you will do a QFD, quality function deployment, is a risk assessment tool. And then on the basis of your quality function deployment results, you will be in a better position to design your facility and your infrastructure accordingly, which is almost a risk-free design. So the design, this QFD design or risk-based design is totally addressing the current GMP standards as well as the possible risk associated to your investment, to your equipment, to your personals, to your processes. So then you go to the regulatory evaluation of the design, whether your design complying the local or the international regulatory standards or not. Then design approval from regulatory, you submit the design. This is also, this facility has been provided by FDA also, which is called, I think, FDM 135. So FDA is also providing the facility to pre-approve the design, pre-assess the design. So when you are not, when you have just developed your design, before laying the first brick or first foundation of your facility, you can send the design to the FDA, and FDA is going to evaluate your design against set money, and then it send back the design with his observation that whether the design fulfills the regulatory requirement of FDA or not. But this assessment of FDA is not giving you any guarantee. Definitely, it facilitates you, or it gives you an approaching picture that you will be approved by the FDA, but it will not give you any guarantee that after completing your project, the FDA has no objection on your project. So after completing your project, the FDA again officially assess your facility, and it has total rights whether to approve or disapprove your facility. So after the regulatory approval, you will be having the budgeted design, that is BCWS, budgeted cost of work schedule. Then the next phase is to budget your infrastructure, that how much you are investing on equipment, how much you are investing on gray structure, how much investing on your processes, how much you are going to invest on the utilities. So this is all estimating the budget, and then estimating financial engineering based on local economic stability. Then it contains your financials of marketing. So how much finances you are allocating to marketing and sales. After estimating the budgets, estimating the operating cost, then the next step is the OPEX. This is something which is pertaining to CapEx. This is OPEX, that how much operating cost you are going to incur on your processes. Then estimating product life cycle management. Product life cycle management means that there are two critical parameters. That first critical parameter is that the products you are manufacturing to the plant, how long it will be lasting in the targeted market. For example, if you are developing some cephalosporin product, so in Pakistani market, so the question is that you must know that how long your product will survive, this specific product will survive in Pakistani market. Or if you are targeting the Far East market, or you are targeting the Central Asian market, so you must know that how. As well as the second aspect is that how long your investment means your infrastructure, will be compatible as per regulation. So if your infrastructure, if you think that with the regulations are changing very dramatically, so the time of investment is relatively lower as compared to the regulations which are almost stagnant, which are not changing with rapidly. So it's very important that the life, you must know while you're doing the investment, you must know the life of your plant and the life of your economic value added is again economic parameter, benefit to cost ratio and break-even points. And then on the basis of all such data, you will be able to decide the feasibility of the project. If your project is feasible, then hiring the project team, management team, and kick off the project. If your project is infeasible, then explore the other markets where changes of feasibility are high, and data analysis, then again do the data analysis feasibility for other markets and make the initiatives. So it's all the typical process flow of pharmaceutical manufacturing plant. I got, you don't, do you have any point to, or you question, then you are always welcome. Oh.
Sir, I think we should continue with the presentation. We'll take the questions at the end.
Okay, no problem. Okay. So it's simply a more simplified form of process and facility design, including all supports like HPC, water system, material traffic control. First of all, you have to capture the URS, user requirement specification or customer needs through marketing research or market analysis. Then the data you gather from the market should be analyzed by your analyst. Then, on the basis of the data, you estimate the capacity of your plant, the capacity of your equipment, the area of your plant, the capacity of your utilities like HVAC and water system. And then, after estimating the capacities, you go to the plant layout. So, plant layout is something, is a drawing of the plant where you can accommodate this production capacities in compliance with the local regulatory standards or the regulatory standards in which to which you are following. Especially in Pakistan, we are following the Schedule 2, which is the release of the draft. But in US, it's 21 CFR is a release of FDA. So or in India, we are following the Schedule M. So these are all the regulatory standards. So where the plant is situated, and it depends upon where the plant is situated. So you have to follow the local regulatory standards and to which markets you are targeting. So if you are targeting the Kenyan market, or if you are targeting the Far East market, so you have to follow the regulatory standards and compliance of that market you are targeting. So equipment selection, select the best equipment based on URS. Plant DFM, design for manufacturability. So it is again a sort of a risk assessment, pre-risk assessment. So this is called something that you develop a plant based on the available resources. So design for manufacturability is something pertaining to the selection of resources which are available in the market. So you, it makes sure that you are not listing any resource which is unavailable or is odd in the market. So process risk assessment. After doing all this, you develop the processes and doing the risk assessment. This is something pertaining to quality by design. So your process is spent based on the compliance of CGMP and compliance of economics of quality. Means the feasibility or viability, business viability. So in regulatory compliance, the prominent agencies which are responsible to regulate the pharma business are DRAP here, which released Schedule B2. FDA is the custodian of 21 CFR, and ICH, International Council for Harmonization. And these ICH are issuing the Q prefix guidelines for CGMP. So come to something, a micro level of process design, how the process is designed, how we design the process and how it works. So there are two critical features. Collectively exhaustive feature. This is collectively exhaustive feature. And another one called mutually exclusive feature. So what is mutually exclusive? For example, here's a very simple example. You toss a coin. So the coin result either head or tail. So this option is called mutually exhaustive. Mutually exhaustive means that you have various options and you select only one out of it. You cannot select more than one. So this is called mutually exclusive options. This is figure as showing the risk-based strategy for cross-contamination control. You are up now. I'm deciphering the whole paragraph. The process design should be made keeping the optimum trade-off of mutually exclusive isolations between the two products of opposite therapeutic effects being manufactured at a time, same premises. For example, you have the same premises and you want to manufacture the tablets. One is for blood pressure Blood pressure means there's something which is like a blood thinner, and another one is for
Philosophy of process design.
blood coagulator. Blood coagulator is normally given to where the people are having the cut on their skin, so that the tablet or the therapy is used to coagulate the blood. So these are the opposite products, and definitely these opposite products are manufactured in the same single facility. So what will be your strategy? Because the contamination of these two products with each other is giving you a very devastating results. So somebody who is under the heart attack, if he is taking or she is taking the blood coagulator, then his blood clogging is increasing, and vice versa. So how you can manage these products in a single facility? So this is called mutually exclusive. This These are variations. For example, a single facility with very clear segregation. This is theoretically possible, but you cannot make sure that the mix-ups are not there. This is not collectively exhausted, mutually exclusive. There are two different facilities and they are operating independently. This is one of the, again, one of the very ideal picture, because it is sometimes not financially or economically viable that for each product you develop a separate facility, until and unless there is a regulatory obligation, like for hormones or like for aerosols or like for cephalosporins. Again, the most applicable scenario is the scenario S, where A and B products are manufactured in a same facility, and this overlapping region is a region of risk. So this is the region where you have the maximum probability of cross contamination. So you have to address this region in order to make sure that this region must be depleted or must be eliminated, and convert the whole process from scenario S to scenario P. So scenario P, in a single facility, when you are developing manufacturing two opposite therapeutic forms, then scenario P is the ideal. So you are dragging scenario S into the scenario P, and this is the best condition for the design of the facility. So you are focusing scenario P for the designing at the moment of you are designing the facility. But in real life, it is lying between P and S. So the lesser this overlapping region will be, the more you are complying with the contamination control strategy. This is the basic philosophy of the process design of any pharmaceutical industry or manufacturing plant. So you normally, I guys, if you please mute your mics. There are two types of projects. In the emerging economies, for example, if the economic condition is very good in a country, then you generally opt for the greenfield project. Greenfield project, that means that you are starting the project from the scratch. You have the piece of land, and you are developing your pharmaceutical plant on a green field or on a raw land, and then you have very good opportunities for regulatory compliance, because there are no restriction, there are no constraints, no hardware constraints, and nothing. So you have the carte blanche to do anything you want in order to comply the regulatory standards and in order to comply your financial standards or your plant standards pertaining to the capacities and pertaining to the automation of your plant. But on the other hand, if you are living in economies like Pakistan, where there is economic recession, economic shocks all the time, so your capitalization on existing or legacy facilities. So most of the people in Pakistan or most of the investors in Pakistan are trying to extend their facilities, their existing facilities. So extending the facility is really a big challenge until and unless the facility is initially designed for extension or expansion. Like I told before earlier that the facilities which is based on the flexible panels or clean room panels has the capability to expand in the future. But the facility which is initially constructed on a rigid infrastructure like masonry blocks uh, or RCC, then it's really difficult. But in reality, especially in Pakistan or the economies which are weaker, the people are normally accommodating their new molecules into the facility which are already exist. So this is a this is a very big challenge, and this is called a brownfield project. It's not easy to incorporate amendment and legacy. Legacy facility is the facility already built, the old facility, as the regulatory compliances are often outdated because you developed the legacy facility, for example, 10 or 15 years or 20 years before. So it's really difficult for you to implement the current regulatory standards or current GMP to the legacy facility. So legacy facilities are generally modulated in a longer period due to higher sales demands and questionable compliances of CGMP. There are always these two challenges because sales guys are always asking for the higher volumes over the higher outputs to sell their products, while your infrastructure does not allow to you to expand easily. Hence, poorly managed. So, when you are screwing the facility and when you are crimping everything into the facility, then the facility will be poorly managed, and you come up with various compromises on regulatory compliances and the quality of your product. But most of the time, Pakistani market is facing the brownfield project. They are often labor or knowledge intensive in nature. So starting from the R&D, design R&D, for example, you want to develop certain product. The latest examples like sitagliptin. Take an example. This is a new product. So how you develop the sitagliptin is a generic application. The product's already developed and is already in the market. So what you are going to do, you are replicating the generic product. So it is how you develop the generic product. You just follow the rules. You just mix up the drug substances, and then following processes, you will come up with the drug products. So the most smartest way of doing the R&D is now with the DOE, design of experiment. Design of experiment is a statistical tool that helps identify critical variables and their interactions, allowing scientists to make data-driven decisions. So, as is a statistical tool, so it allows you to to take a data-driven decision. It's very powerful statistical tool, design expert. So I don't know, but in US, when I met the pharmacist of the US, they have the experience of working on design expert. This is a tool is a software based on design of experiment, and this design expert is especially very effective tool for generic product development or even for the innovation of new product. So design expert is one of the examples you generally come across during the Doctor of Pharmacy programs. This approach minimizes trial and error. So how it gives you a leverage. For research and development, the best leverage is to reduce the number of trial and error, the number of trials. So this design expert tool, which is based on the design of experiment, this approach minimizes trial and error and reduce development time and ensure robust and reproducible results. So these are the advantages of design expert. Now we see how it works. This is one of the case study. These are the various factors. There are, I think, 5 factors. Concentration of API, milligram, type of excipients, compression force during tablet compression, granulation method, and duplication amount. So there are 3 levels. It's just for the sake of example. 50 milligrams, 100 milligrams, 150 milligrams of API, types of excipients, lactose, microcrystalline cellulose, starch, compression force these are the compression force, wet granulation, dry granulation, and lubricant amount, which is 0.5%, 1%, and 1 point. So you are combining what design expert is doing. You just give the factors to the design expert. You just give the levels. And here there are 3 levels. You can go up to 20, 30 levels, and then the design expert during the R&D phase, and then the design expert will give you the best trade-off, the best combination of factors and their levels. So sometime you may come up with this factor of concentration, for example, 150, a type of excipient, for example, lactose, a compression, for example, 10. So it can be any combination. So design expert as based on statistical data analysis. So this tool is giving you the best combination for your product. So I generally recommend the design expert tool for all those guys who are working in the R&D or product development in Pakistan. So now come with the conventional design versus quality by design. Conventional design of the processes, we are focusing the compliance focus. We are focusing the fixed regulatory standards, while the risk-based design process, which is a QBD approach, considering possible process risks. So it is not fixed. Here we are just following the law, following the regulation here. In addition to the following of regulations, we are also doing the possible process risk. We are also considering the possible process risk. मैं इसकी एक example आपको दे देता हूँ. It's a very simple example. For example, a toddler is sleeping in a bed. Why you are putting Gautakia at the side of the toddler? Because when he turns or she turns, the Gautakia is making sure that he will not fall down. So it's simply a risk-based design. So this is a very simple example. Conventional design is based on fixed parameters like GMP. The risk-based design is based on the dynamic parameters like CGMP. The current good manufacturing. So the risk, if you are designing your plant on the basis of the CGMP instead of GMP, then your plant will have the capability to accept the flexibility of change or to accept the change in the GMP. So what you are putting here, you are putting the flexibility in your plant. In conventional process, you are focusing on end product testing, which is done by the QC. In risk-based process design, you are more focusing on process control. So you make sure that the processes which are giving the final output should be tuned up in such a way that it will give you always the good product and acceptable product. So, in conventional process, your plant is less flexible and less ability to incorporate changes. In risk-based design, your plant will be more flexible and ability to adapt changes. Scale-up challenge. So, after completing the R&D, when you are going to the commercial manufacturing of your product, then there are two very important things, which is one is repeatability and reproducibility. Repeatability is something pertaining to the processes, something pertaining to the processes, and reproducibility is something pertaining to the product. So you make sure that your processes should follow the same steps. Your processes are accurately tuned up, and they are producing the same product, drug products, or same results at the end of the processes. So the scale up challenges, the two more prominent scale up challenges are repeatability and reproducibility. Another thing is the CCS. CCS is contamination control strategy, which is the outcome of the Annexure 1. Annexure 1 is a document released by ICH in 2022, and this document is widely acceptable by all the regulatory agencies, including FDA. Then fourth thing is process optimization, how you are fine-tuning your processes and digitalization of the processes. There is something pertaining to CFR 21 CFR 111. QC versus QA. QC is a reactive approach that filters out the defective drug products. QA is a proactive approach that prevents to produce the defective products. So it prevents the processes to produce the defective product. So one is a reactive approach and one is a proactive approach. Annexure 1 in 2022, ICH released a document, Annexure 1, which provides detailed guidelines on manufacture of sterile products. So initially the Annexure 1 is dedicated for the sterile products, which are, so it makes sure that the sterile products are free from any microbial contamination and any viable, non-viable count, pyrogen free. So this is initially the purpose of the document, but now the document is widely acceptable and applicable to both the sterile and non-sterile products. Contamination control strategies, a risk-based approach evolved through QBD, quality by design, that includes how it works. It prevents preventive measures against possible contamination. It gives you the liberty, it gives you the directives to design your processes such that there will be zero contamination during the processing. Number two, it gives you a risk management associated to contamination. It gives you all the possibilities that how the contamination can distort your product. So all the risks pertaining to the contaminations are already addressed in the contamination control strategy. Is a holistic approach to ensure the organizational synergy. So every concern of the process or every custodian of the process must have a very clear idea how could he or she deliver his or her energy to comply with the contamination control, are not are compatible with the CGMP and are free from any contamination. So any possible contamination which is which can induce during the process can is addressed in the holistic approach. Continuous monitoring. So continuous monitoring is a part of the continuous manufacturing, in which instead of doing the random sampling, the product or the in-process material is being continuously monitored by the sophisticated technology. Generally, this technology comes under the PAT, process analytical technology, and this makes sure that the product safety, quality, and efficacy will be intact during every stage of the processing. So come back to Pakistan. Common challenges in legacy facility. There are you have the legacy facility, or you have the existing facility. So what are the challenges you are come across, especially while you are facing the change in the regulatory standards? So the policy is policy. So the challenges are number one, cautious circulation is an uncontrolled movement within the controlled areas. So the area, pharmaceutical area, whether they are classified or unclassified, when you enter into the plant, it is considered a controlled area. So every area is a controlled area. So when you are cautious circulation or uncontrolled circulation in the area, it is one of the biggest challenges, because the plant is not designed to address that issue. So most of the plants in Pakistan are not designed to address the uncontrolled movement of personnel and material in the area. Number two, the lack of unidirectional material flow, crossing of materials, equipment, and personnel flow. Number three, excessive handling of material, unnecessary handling of material, which give you unnecessary cost and GMP burden, and the risk on product contamination. Inconsistent gowning practices. So in some companies, there's very poor gowning practices. So these practices are the challenges. They are the one of the challenges in the legacy facilities. Inadequate segregation of unit operations and drug substances. So most of the legacy facilities or older plants are incapable to do the segregation of operations and incapable to comply with the contamination control strategies. Inefficient organization of environment or hygienic zones. Hygienic zones are the HVAC zones. So most of the plant exist, which all developed 20 or 15 years ago, does not have the enough capability to maintain the hygienic zone requirements of today's GMP. Product contact part risk, including lack of non-reactive, non-additive, and non-absorptive product parts. So if they are deploying the old or obsolete or outdated equipment, so the outdated equipments have these three challenges. The product contact parts are non-reactive. These challenges are that the product contact parts must be non-reactive, must be non-additive, and must be non-absorptive. But the obsolete equipments does not comply with this. Sometimes you will see the rust on the machine. Sometimes you will see the erosion on the machine due to with the reaction of the powder. So all such things must be addressed in a contamination control strategy, and your equipment must be selected in such a way that its product contact parts must be non-reactive, non-additive, and non-absorptive. So this is hygienic zone. Hygienic zone. Always remember that the cleanliness concept in pharmaceutical industry always comes from the two sources. One is the human practice, personal flow, and number two is through HVAC. So cleaning of an area is always based on personal flow or personal practices, including gowning, degowning, and handling of material, activities of a person, and primarily based on HVAC. So HVAC is heating, ventilation, air conditioning. You maintain the cleanliness of a particular area through air pressurization, air changes per hour, and air filtration. So this is a typical protocol of the pharmaceutical area. CHC means the area which is controlled but not classified. This is class D area where you generally doing the secondary packaging. This is class D area where you are handling the product, which are generally taken oral dosage. This is class C, is a sort of the sterile area. Class C is sterile area. Class B is the area generally experienced in the labs. And class A is the most sacred area. This is the area under lab. So this is a typical sequence of protection. So you cannot maintain the class A area around the class D area. So for the class A area, if you maintain the class area around class D area, then it will be increasing the risk of contamination and cleanliness for class area. So it's a protocol that the most important area, the cleanest area is surrounded by the less clean, and then less clean, and then less clean, and then less clean. So this is a general protocol of the HVAC or cleanliness classification.
Sir, I'm so sorry. Sir, I'm so sorry for intervening. Can you please just speed it up a little bit, because we are running out of time and there is specific stuff.
Okay. Clean room standards.
I'm so sorry for cutting you.
No problem. There are the clean room standards. You can read the clean room standard 14644-1. So there are complete clean room standards available for particle viable and non-viable particle counts. So normal versus potent drug substances, there are different protocols available for the normal drug substances and for potent drug substances. HBPL is a human-based exposure limit, and for category 1, it is a least potent category, and category 4 is the highest potent category. Well, and at last, after completing the plant, you will go to the process validation, the design qualification, the process qualification, or continuous process verification for continuous manufacturing. And that's all from my side. Any question, I'm here to answer. Hello?
On behalf of all of our attendees, thank you so much for the insightful session. Your insights, experience, and valuable knowledge made this session both informative and inspiring for all of us. I know that many participants, including myself, will walk away with valuable knowledge that you shared today. The session is open for questions now. We already had one question in the conversation box. I would like to address it first. It's by Dr. Faryad Ashraf, and she's asking that does design expert
Yumna,
also help?
कमेंट में दे दूं अतीक साहब अस्सलाम वालेकुम
वालेकुम सलाम कमर साहब आवाज से पहचान लिया मैंने
जी मैं अतीक साहब मैं यमुना ने आपसे स्लो डाउन करने का कहा था बिल्कुल खत्म करने को नहीं कहा था आपने फौरन खत्म कर
नहीं बस इतना ही था क्योंकि यमुना पहले बता चुकी थी कि वो 1 घंटे का सेशन है
जी नहीं सर वो मतलब ये तो आपके लिए बहुत थोड़ी सी चीज है वरना मैं तो समझता हूँ आई थिंक दैट यू कैन हैव अ लॉट ऑफ थिंग्स और डेज एंड डेज इसके ऊपर डिस्कशन हो सकते हैं यू हैव सो मच नॉलेज अबाउट इट और यहाँ मेरा ख्याल है कि बहुत कम लोगों को मालूम होगा कि ये माहिर इकोनॉमिक्स भी हैं अतीक साहब इंजीनियरिंग के साथ साथ इकोनॉमिक्स के भी बहुत बड़े माहिर हैं
जबरदस्त
जी प्लीज यमुना.
I'm really sorry to cut you short, but हम थोड़ा सा time में थोड़े से bound हैं, so I really apologize. Sir, आप please अगर थोड़े से questions address कर दें. एक question से क्या आया है, what is the role of pharmacist in this regard? Answer please.
Pharmacist basically is a product custodian. Pharmaceutical product है, तो pharmacist से बेहतर कौन जान सकता है, खासतौर पे जो इंडस्ट्रियल फार्मासिस्ट होते हैं, तो वो बेसिकली उनको पता होगा कि प्रोडक्ट हैंडलिंग के प्रोटोकॉल्स क्या हैं. बेसिकली किसी इंजीनियर केमिस्ट को इतना इसका इल्म नहीं होता जितना फार्मासिस्ट को होता है. कंटामिनेशन, क्रॉस कंटामिनेशन की वजह से जो लैक ऑफ थेरापेटिक इफेक्ट्स होते हैं या एडवर्स ड्रग रिएक्शंस होते हैं ड्यू टू कंटामिनेशन, वो फार्मासिस्ट को पता है. तो जो ग्रेविटी है किसी भी कंटामिनेशन की या किसी भी प्रोडक्ट की हैंडलिंग की, वो फार्मासिस्ट से बेहतर तो कोई जान ही नहीं सकता. दैट इज व्हाई फार्मासिस्ट इज प्लेइंग अ वाइटल रोल इन द प्रोसेस डिजाइन.
Sir Kamar, if you would like to have an insight on today's session, I would be grateful. हम फिर इसको conclude करेंगे.
Yes, please. I think it's been a long time. So, and he is also not well. I don't think we should put some extra, exert some more pressure on Atiq Saab. He is sick actually. So it would be better to conclude the session.
Thank you. Thank you so much, sir. Thank you, Sir Atiq, for your precious time, and we always look forward to having such sessions. हम आपकी expertise का आगे भी इंशाल्लाह फायदा उठाते रहेंगे. And thank you once again.
बहुत शुक्रिया. अस्सलाम
वालेकुम. Thank you so much for all the attendees for attending this session. We are concluding now. Thank you so much. Allah Hafiz.